Hello everyone, I am Kamalesh Damodharan, founder of ScientiFlow. Today I will show you how to perform protein-ligand molecular dynamics simulation using ScientiFlow. Let's get started. First step is to prepare the samples for performing protein-ligand MD simulation. For demonstration, I am going to have three docked complexes, hit1 complex, hit2 complex and hit3 complex.pdb files. I am going to use Chimera to prepare the samples. You could use any of your favorite visualization tool like PyMol or VMD or as you wish. For the demonstration, I will utilize Chimera. For performing protein-ligand MD simulation with ScientiFlow, we need to split the protein and ligand files into two separate files from the docked complex. Here we do not change the orientation of the protein or ligand, we are just going to select them and save them into two separate files. For doing this, we simply have to navigate to the select and then select the protein chain. Now we have to press the file, save PDB and then type the desired name. In my case, I am going to save it as hit1 protein only.pdb. And don't forget to check this box which will allow to save only the selected atoms and then click save button. Now we have to select the ligand alone. So for doing this, go to select chain and then heteroatom. Now only the ligand atom is being selected. Now again click the file, save PDB, type hit1 ligand only and then check this save selected atoms only. Likewise, repeat this process for all the two other complexes as well. Once we separated the protein and ligand files from all the docked complexes, head over to the ScientiFlow and then head over to the file manager. Simply click the file upload button and then navigate to the desired folder and then upload all these sample files. Now it's time to configure the pipeline for performing protein-ligand MD simulation. Head over to the pipeline section. From the top, if you see, the pipeline splits into two directions. One is for protein-ligand MD simulation, another is for protein alone MD simulation. This is a case for apoprotein where you would like to understand the impact of ligand on the protein's conformation. In that case, you will be toggling this on. For our case, we are going to work with protein-ligand MD simulation, so I am keeping this on and then keeping this off. Now I would like to choose the force field. To get a help about the selection of the force field, you can simply click this one and read this documentation to understand. For this study, I am going to use CHOM36 force field and all the steps for ligand parametrization specific to the particular force field will be executed. Towards the down, we will have a choice to select the shape of the box. Again, we will have the tooltip to help us. And I am going to choose cubic box as a default one. And subsequently, the solvation steps and addition of ions, energy minimization, equilibration of NVT and NPT and production MD run, everything will be handled automatically. There is one thing you need to change further is, you can choose to perform the simulation with CPU only or GPU mode. In my case, I have only CPU, so I am going with this one. If your workstation is equipped with GPU, toggle this on. Since I have only CPU workstation, I am going to activate this one on. Now the commands for specific to the CPU will be executed. And finally, MD simulation results can be toggled on and off. For my study, I would like to perform all the analysis except SASA and DSSP. So I am just toggling these two off and rest of them I am keeping them on to perform the simulation. After this, we just simply click the save button and then save the whole configuration. Now head over to the job section. Now we will submit a job using the particular samples that we uploaded and then perform the MD simulation. Let us create a job. The title is something that you can recognize from this job. I am going to say hit1 MD simulation. This is something I am going to recognize. The job directory is something that which will be created inside the while executing the project. So keep the job directory unique for each complexes. In my case, I am going to keep hit1. This will prompt that job directory is available, meaning that you have not created any other jobs within the same name. Click continue, and then you will be prompted to select the start and end node of the simulation. In my case, I am going to start the simulation from the head and then take it until the down, completely after the analysis. Imagine the scenario, if you wanted to perform only until the end of the production run, you can stop the simulation right here. But in my case, I am going to include even the analysis part, so I am going to run the part until the end of the simulation. And then click continue. Now we need to configure the environment variables. Environment variables are nothing but the dynamic variables. Here we just going to provide the details of the protein and ligand. For instance, protein file no extension means we have to provide the name of the protein file without extension .pdb, which is nothing but the protein and ligand files that we have uploaded few minutes back. In our case, it is hit1 underscore protein underscore only. And for ligand also similarly, we need to provide without the extension. So I am going to write hit1 underscore ligand underscore only. Third, we need to give the nanosecond timescale in nanosecond. I am going to keep this 100 as it is. The fourth one is not going to be relevant at this moment. This is for data visualization time. At this time we will come to this particular row. And fifth one, it is just number of cores. I am going to keep it 8 as it is. If you wish to change, you can change. Now we will just going to prompt with server name that we have configured already. We just leave it as it is and then click the submit job. Repeat this process to submit the job for all the 3 hits. Now we have submitted job for all the 3 hits. Before we proceed to execute these jobs, I hope you all have completed the getting started process, that is 3-step process as indicated here. If you have already completed this process, simply head over to the terminal and type the command ScientiFlow CLI flag help. This command will list all the commands that is available from the ScientiFlow CLI for your reference. Now I am going to run the command ScientiFlow CLI flag list jobs. This command will list all the pending jobs that has been submitted by you in the ScientiFlow application. To execute a particular job by ID, you just have to execute this command, that is execute job by ID. I am going to call the ScientiFlow CLI and then run the command execute job by ID, and then you need to type the job ID. In my case, it is 520. Once you press enter, it will pull all your sample files that you have submitted to the ScientiFlow application, and then it will also download the softwares required for you to run the workflow. So these softwares that has been containerized and then it will be shipped directly to you, so you do not have to install anything manually. Since it has been already downloaded in my case, it is just using the cached image. Now it is automatically started performing the MD simulation. So protein parameterization has been completed and then ligand parameterization is running now. So it will automatically perform the ligand parameterization and that is also completed. And then it is subsequently adding the solvation of the water molecules and then adding the ions. Every step is carried out automatically. Now it is automatically performing the energy minimization. Now it is performing NVT equilibration step. Subsequently, it will run for NPT and MD and all the analysis as well. Now, if you would like to see your simulation files, you just navigate to the directory where you set the set base directory in your terminal. So in my case, I am already inside the folder. Look for the project title. So here it is demo. Demo is the project name I created. So I am finding the demo folder here. If you click here, we will see the project job directory. So in here, in this case, we have hit1 as the project job directory. If you navigate to that one, you will find the simulation that has been carried out for the hit1 ligand and hit1 protein complex. So you will find all the simulation process has been carried out, and then a NVT step is currently running. So all the simulation steps until the NVT is available here. Now let us run the simulation for all 3 hits and wait for the workflow to be completed for all 3 hits. Now the workflow for all the 3 hits has been completed successfully. It means we have completed the production MD simulation and also the analysis part as we were executing in the previous step. Now in order to generate the publication ready images, simply head over to the pipeline section. Navigate towards the right for identifying the workflow related to the publication ready images, and then toggle on and toggle off whatever the analysis you would like to be visualized. In my case, I am going to toggle off SASA and also ligand RMSD just for the demonstration. After this, you just have to save this changes and then navigate towards the job directory. Now you could just create a new job for visualizing the data. Type your job title. Results visualization I am keeping. Type the job title as you wish, and then job directory, results. This is the place where all our images will be stored, and then navigate towards the right for identifying the place for the workflow. I am just choosing my start node and then end node. This is the environmental variable. Here these 3 columns can be ignored as it is. Do not disturb them. Just modify this particular column. This column will correspond to all the project folders names here. Hit 1, hit 2, hit 3. I am just going to give all these folder names to visualize them in the plots. And then click submit job. Now simply open the terminal, look for all the pending jobs. I am typing ScientiFlow CLI flag list jobs. Since we have submitted only one job and it is in pending, I am just going to run this job by ScientiFlow CLI flag execute job by ID and then ID. That is 523 project job ID. If I run this command, it will start pulling all the relevant files and folder required to run this data visualization. All the software has been already cached. It is already generating the plots for RMSD, RMSF, RG, H2 bond and GMXMOPBSA. And all these images has been generated successfully, and now we can just navigate to the folder. We see that there is a folder being created, results. If we just open, we are seeing the results already there. GMXMPBC results is there, and then hydrogen bond, radius of gyration, RMSD, RMSF. All the analysis that we have toggled on has been generated here. I hope this tutorial will help you to run the simulation of protein ligand and also generate the publication ready images for your study.